The overall goal of this procedure is to induce the conversion of germ cells to neurons in C. elegans.
This method can help investigating the implication of biological processes such as signaling pathways or epigenetics in the regulation of cellular fate reprogramming. So the main advantage of the method described in this video is that in living animals, germ cells can be challenged by the overexpression of a fate-inducing transcription factor, while in parallel, a genetic screen such as an RNAi screen can be performed in order to identify novel factors that are implicated in regulating cellular reprogramming.
The implications of this technique extend toward regenerative therapy because this in vivo method could be used to study cellular reprogramming under different developmental and environmental conditions.
Visual demonstration of this method is critical as the reprogramming steps are difficult to learn because the germ cell to neuron conversion phenotype upon depletion of Lin53 only happens in carefully controlled conditions.
For the RNAi plates, load 6 cm plates with NGM agar containing IPTG and carbamazepine. Let them dry for 24 to 48 hours at room temperature in the dark. Then transfer them to 4 degrees Celsius for up to 14 days. Next, streak selective plates containing carbamazepine and tetracycline with RNAi bacteria clones with L4440 plasmid carrying the Lin53 gene. Use a 3-phase streaking pattern and be sure to plate an empty vector control. Then, grow the plates overnight at 37 degrees Celsius. The next day, pick at least 3 single colonies